So yeah, the question that my lab is really excited about is, can the body undergo morphogenesis again as an adult? So like, why do you need like a liver transplant? If you grew a liver when you were in your mother's womb and every single cell in your body has the same DNA, why can't you just reactivate that same program when as an adult? And the newt is a wonderful, beautiful example of that. So this is a time-lapse video of actual regeneration of a newt's limb. It's just a miracle of biology that you can have this dedifferentiation event and a recapitulation of embryogenesis and regrowing all the same structures. They also try to, and this is a cartoon that's trying to illustrate that it's not one stem cell that does it, but it's the coordinated activity of multiple separate stem cells that are orchestrated by this structure called a blastema at the end of the womb. Oops, sorry about that. Okay. All right. So, and then this is trying to depict like the red is like a mesenchymal stem cell, white is like a bone stem cell, and green is like an epithelial stem cell. And then together they're going to kind of come together to recreate that tissue. And I love this biology so much. I'm trying to teach myself how to do acrylic painting. And this is, you can trick a salamander making a supernumerary limb. It's just by doing some skin surgery. So if you do a rotation flap, so you have positionally mismatched cells. So you basically take a piece of skin and rotate it. So now the cells next to them work where they started. And then you reroute a nerve. That's enough to trigger the newt to making a whole new limb, where it shouldn't make a limb even, which is pretty cool. Okay. All right. So if we can't regrow a whole limb now, can we at least regrow a mini appendage? They call hair follicles appendages. And so I'll tell you a story about that today and a story about our commensal bacteria that live in our skin. So it turns out that hair follicle development and limb development are very overlapping. So at the top you have the effects of beta-catenin mutations. And in those mice, you can notice they don't have many, they're losing their limbs and their tail development. And that's a severe loss of function. If that same mouse has a minor loss of function, they can grow past that point, but they don't develop hair follicles. And the same thing happens with P63. If you have a mutation in P63, those mice also don't develop limbs with a severe hypomorph. But then when you have a less severe mutation, they can live past that event, but they don't make hair follicles. And so if we can learn about how to make a new hair follicle, maybe we can learn about how to make a new limb. So the question is, can we make new hair follicles? And you certainly can from stem cells. So this is kind of a fun experiment I did when I was a postdoc. I just took a solution of a keratinocyte stem cell and a fibroblast stem cell. So it's just a cloudy solution. And if you inject it under the skin of a mouse, it's kind of like the Terminator. Did you guys ever see that movie, The Terminator, where he's like a pool of something on the ground, and he can self-assemble himself? Well, that's the cool thing about stem cells, is they can self-assemble. So even though it's just a cloudy solution, if you inject it, they make skin. They basically make a cyst, and then within that cyst, hair follicles develop. And here, I just cut open the hair follicle. And you can, I cut open the cyst rather, and you can unfurl the hair that kind of grew in the middle of it, which is pretty cool. All right. And you can also do that. You can make the hair look more normal. So this is like a nude mouse, and you can do grafting in chambers. And if you put the right stem cells in the chamber, you can make new hair follicles as well there too. So what about, that's great. You can do that in vitro and kind of with purified stem cells, but does this normally happen in vivo? Like, can you make a new hair follicle? And so when I was in training, not super long ago, I guess, about 20 years ago, I was taught when I was a resident, you could not make a new hair follicle. It was just too complicated that mice and humans can't do it. But it turns out that, and the reason why they thought that was, it's really complicated. There's at least 3 different stem cell populations in the hair follicle. There's 10 different epithelial lineages. There's multiple types of tissues, like mesenchymal tissues, like fibroblasts. And adipose cells. So anyway, people thought it was just too complicated. But it turns out that in the 50s, they'd done this. So I joke that I don't do any innovative work because all the work I did was started in the 1950s. But this guy, Charles Breedis, is doing these big wounds in the back of a rabbit. And at the very center of those the wound would heal, and they would pull off all that new skin, and they would do a whole mount prep on it. And you can see at the edge, there's no hair follicles that form. But in the center, you see there's all these new hair follicles the rabbit made. Basically, we in the lab that I did my postdoc in, this other talented postdoc, Mayumi Ito, discovered that this actually does happen, and we can see it in mice. And so if you do a wound in a mouse, like a large 1 by 1 centimeter, it closes, and first you just see a scar. But then with time, you can see there's new hair follicles that form in the center of the wound. They're white because the melanocyte stem cells don't migrate into the wound, interestingly enough. And if you look at it by histology, you can see all the same processes during development. So just like a lot of organs form, you can see this epithelial placoid and hair germ forming that turn into a peg and then have a fully formed hair follicle. And if you do like a whole mount, you can see the same thing. So it's just like Charles Breedis did that as an original studies on rabbits. If you look at mice, there's this like keratin 17 positive staining on the periphery where you have hair follicles, but in the middle there's nothing. But with time, you can see these new dots forming. Like, so these mice are making new hair follicles in the middle. So the question that my lab wanted to ask was, what regulates this and how can we induce this? Like, what are the earliest events in wound healing that can reactivate these developmental programs? And so we did a screen of mice that we knew did it really well and mice that do it really poorly. And basically, I'm not going to go over all the different studies we've done. So we have like an alphabet soup of a lot of different molecular pathways that are turned on to make this happen. But in common, what we notice is they all cause decreased differentiation and activation of the Wnt pathway, which is known to stimulate it during development. But we noticed something kind of interesting. At all of these different models, we looked at the gene expression categories of what was associated with good regeneration. And the really interesting thing is, for a lot of them, we saw Staph aureus infection. So here's one. Here's another one for the RNA cell knockout mouse that Charlie's working on. This in the audience. That one has a lot of extra hair follicles. And when we looked at what was upregulating that, we see this signature for Staph aureus. And so we were curious, well, why does Staph aureus infection associate with regeneration? Like, how do bacteria in the microbiome, how do they interact to promote regeneration? First we did is we just grew up some mice in a bubble, like the bubble boy, where they don't get exposed to bacteria, and we did wounds on them, and they don't develop hair follicles. So they didn't regenerate very well. And then we took those same mice that grew up in the bubble, and then we took them out of the bubble so they could be happy and live with non-bubble people. And they found that, and we could rescue their regeneration. So when you took the mice out of the bubble and lived with their non-bubble friends, they can, or even siblings, then that can rescue their regeneration. So they get good regeneration. And then we found out that you can actually even just change how much bedding changes you do. So the cool thing about the mouse house is they have their own like cleaning service, and in addition to their own catering and their own veterinarians. And you can ask the cleaning people to do less cleaning of their cages, and so their cages get dirtier. So when we asked them not to clean their cages, the mice actually regenerated better. And you can do a lot of fancy genetics to show that the diversity of the bacteria is higher in all these different conditions, just confirming that there's more bacteria. So then we said, well, can we add antibiotics? So we added some antibiotics to the mice, and so they have less regeneration if you add some Neosporin. So then we said, well, okay, that's all normal bacteria. Can we add extra bacteria and see more regeneration? So we did that. So we added Staph aureus, just like we saw in those gene signature profiles, those genes were associated with higher regeneration. If we add Staph aureus, which normally causes an infection, and we don't add enough to kill the mouse or anything, we can actually stimulate regeneration if we add Staph aureus. So we actually even did this in people, where we asked them to we did identical punch wounds in the back of the people's legs. And on half, we said, on one leg, just put Vaseline. On the other side of your leg, put Neosporin. And we found that the side that got Neosporin had less rapid healing. So the Neosporin slowed the healing. And you can see that on the right, each person is normalized to themselves. And you can see all of our subjects showed a worse healing with Neosporin, because your normal microbiome really helps you heal. So then we said, well, what's the mechanism of this? And I won't go into detail for the sake of time. But basically, we did a lot of transcriptomics to say what are the genes that are responsible for how bacteria promote regeneration. And we compared either that loss of bacteria with the germ-free mice in the bubble, or the gain of bacteria with Staph aureus. We said, what are the common transcripts? And so there's a lot of different genes here, and some of your favorite genes, and some of you guys might study some of these genes, but the one that caught our attention the most, it was different in both conditions, the best was IL-1 beta. So IL-1 beta, you can see, went up really well when you add the Staph aureus, and it also went down when you added the germ, when you put them in germ-free. None of the other genes correlated quite as well as IL-1 beta, although they're likely important as well. We basically looked at IL-1 beta, we said, was it sufficient? So we added extra IL-1 beta, and we can promote regeneration. Here, so you can see when we add the recombinant mouse IL-1 beta, we see more regeneration. And then we could take a knockout mouse, where we knock out IL-1 beta, and we add the Staph aureus, and they don't have any extra regeneration. And this stuff's published, so you can actually check it out in the Cell Host Microbe. And what we try to show is that like the bacteria is like a Band-Aid. And so bacteria is good for wound healing. So the conclusions are that many appendage regeneration is possible. Bacteria can promote regeneration, and IL-1 beta is responsible for that. And I want to thank a lot of the people that helped with this work. Gao Feng did most of it. He's running his own lab now at Nanfang Hospital. Dongwon Kim is at Dongsuo University, and he helped out a lot. He's running his own lab there. Rosie Chen's helped as well. Sashank did. Sashank is a professor here at Johns Hopkins. And Amanda Nelson is running her lab at Penn State. So those are the people that helped out, and a lot of them have their own labs, which is great. So thanks very much.